Dr. Manna, could you please start sharing your presentation? Am I audible?
Yes. Yeah, I'm sharing. It's taking some time.
It's okay, it's okay.
So it's still not working. It's anything problem?
I think you can stop presenting and then try again. Just one more time.
Okay, just.
Can you go to your PowerPoint and start PowerPoint presentation?
Yeah.
Yep. It's working. Now, please just say the PowerPoint presentation. Yeah, no, that's fine. You are on the right page.
Mm-hmm.
And just say the presenting, like, not in the Meet, but in the PowerPoint, the presentation mode. It will be fine. Yeah, it may take a little bit of time, but it is fine.
It's in the
Yep,
presentation.
you are in the right place. Yep, that's all good.
Can you see that now?
Yeah, I can.
Yes,
I can.
Yes,
Yeah,
we can
it's
see.
fine.
Yes, yeah.
Just click on the PowerPoint.
Okay, got it.
Yep, you are good. You are good.
Okay,
Yeah,
thank you.
right. Yeah.
Okay, should we start now? Yes.
Yeah. So, just a minute. Should I start?
Yeah, just a minute. I have to give a little introduction of yours.
Okay, thank you.
So, welcome you all again in the third lecture of today's scientific session. Our next speaker is Dr. Dipak Manna. I would like to give a very small introduction of Dr. Manna before starting today's talk. Dr. Manna, just one thing to mention, there would be a reminder at 3 minutes prior to the end of your talk.
Okay.
Dr. Manna completed his doctoral work from Bose Institute, Kolkata, and subsequently he joined University of Rochester, New York as a postdoctoral fellow. Later, he continued his research in Stanford University School of Medicine. He has worked extensively in different aspects of Entamoeba histolytica throughout his research career. Currently, he has characterized a novel transcription factor that regulates encystation, which is responsible for disease transmission. This transcription factor could be used as a better drug target to prevent disease transmission. He has 12 original research publications and one patent. Dr. Manna recently joined Presidency University with prestigious Ramalinga Swami Fellowship. We are extremely happy to have Dr. Manna today as a speaker, and we warmly welcome him to deliver today's talk entitled Host-parasite Interaction and Stage Conversion in Entamoeba, Progress and New Insight. I request Dr. Manna to please continue.
Thank you, Dr. Mondal, for a nice introduction. And I want to welcome everyone in this webinar. And I know many of you are different part of the world, so good morning, good afternoon, and good evening, respectively. Today, I'm actually very privileged and honored to be for the organizers who allow me to present my research work here in this webinar. And today I'm going to talk about host-parasite interaction and stage conversion in Entamoeba parasite. So Entamoeba histolytica is an extracellular protozoan parasite and is one of the leading parasitic cause of death, which kills almost 100,000 people annually. From its name, histolytica, histo means tissue and lytic means dissolving, so we can understand it has capability to destruct the host cells. Entamoeba is worldwide spread, and it's, as I mentioned, it kills almost 100,000 people annually. An infection of Entamoeba starts with ingestion of mature cyst with contaminated food and water, and trophozoites are released in the small intestine. So Entamoeba has a simple life cycle. The cyst form coordinated cyst, which has a chitin cyst wall, which make it resistant from environmental extremes and is responsible for disease transmission. And the trophozoite form, which are responsible for pathogenesis, which can lead to the amoebic dysentery in the large intestine, and also can infect to the tissue barrier to reach to the colon and different part of the tract where it can make the invasive colitis. It can also invade through the tissue barrier to reach to other organs like liver, lungs, and brain, and can form abscess, which are detrimental. In the large intestine, some of the trophozoites undergo incystation to make mature cyst and pass into feces to transmit the disease to the next host. So the stage conversion from trophozoite to cyst is essential for disease transmission. If we look at the global impact of Entamoeba histolytica, diarrheal disease mostly affect in the developing countries, in Southeast Asia, most of the countries in Africa. For example, if we look at Bangladesh, each children out of 30 dies due to diarrhea by their 5th birthday. Not only the mortality among the children, there is a significant growth and developmental delays which are common in children with frequent episodes of diarrheal illness. If we look at the global stat on Entamoeba infection, almost 500 million people infected worldwide. The good thing is only 10% are symptomatic. That is 50 million, and that is a number also huge, which have the severe invasive disease per year, and which kills almost 100,000 people annually. And we are all now aware about the COVID-19 And up to now, almost 7 months passed, we can consider like almost 19 million people are infected or detected positive. And compared to COVID-19, we can compare per year 50 million. So I want to just make this point that there is in the worldwide, there are a lot of people infected by amoeba, and still it's a global health problem. So how Entamoeba kills the cells? Entamoeba interacts with the host through its gal-gal-lectin surface protein, which recognizes to the receptor galactose or N-acetyl-galactosamine. There are other proteins also, STIRF, CARP1, and transmembrane kinase family protein, which are responsible for host-parasite interaction. Upon interaction with the host cells, Entamoeba, 3 events can take place. One is amoebic trogocytosis, apoptosis, and phagocytosis. Trogo means nibbling or biting. It happens to the bigger cells or larger cells where amoeba cannot engulf completely, so it takes a bite at a time. And eventually the host cells die due to loss of membrane integrity. Some of the host cells induce expressing phosphatidylserine or C1 complement protein outside the membrane, which bind to the calreticulin receptor protein of Entamoeba, which triggered the apoptotic cell death of the host cells. And finally, the smaller cells or dead cells are phagocytosed completely by the Entamoeba. Fundamentally, the amoebic trogocytosis and phagocytosis, these processes are different. However, the PI3 kinase and the C2PK protein kinase, which are involved in actin remodeling in both of these processes, are quite similar. So we can make a simple analogy that amoeba also knows his food. So suppose if you have a bigger fruit like apple, we need to take a bite, and if you have a grapes, so you can just take as a whole. So amoeba also knows it. So as I mentioned earlier that Entamoeba has incestation, the stage conversion, which are responsible for disease transmission. However, very little is known about this incestation process. Here today I'm going to talk about identification of a regulator protein that is a transcription factor, which has important role in this incestation pathway. And in the bigger picture, if we can block this incestation, we can block the transmission of this disease. However, there is challenges. So the main challenge is there is no method to get mature cysts from Entamoeba histolytica. So despite effort from different labs in few decades, last year, few decades, there is no method to get mature cysts from histolytica, and most of the work is done in related parasite Entamoeba invadings. So most of my talk, I'll concentrate on the identification of the target of the molecules in Entamoeba invaders. So RNA sequencing of different developmental time points of Entamoeba invaders was performed, where you can see the trophozoite, different stages of incestation and excitation. And we are looking for a transcript which are upregulated during the incestation time point at 24 hours. And we found out almost 616 transcripts which are upregulated. This is the normalized expression of all 616 transcript. The criteria we choose like low at stove and high at 24 hours. And you can see they have different patterns. So one question we ask, why these genes are regulated during incestation and how? So one possible question for why, because these genes may be required for this particular developmental time points, and how these genes are regulated. One plausible explanation would be the promoter of the genes may have some information. So we analyze the promoter by bioinformatic analysis by using MIM and MAST suite. So basically, we took 500 nucleotide upstream from ATG of this specific gene and did the MIM analysis. What it does, it gives consensus DNA motif. We compare all this motif to all promoter database, which is almost 12,000 promoter of this parasite. And by MAST analysis, we annotate those and calculate the frequency and the occurrences. So we ended up with 9 motifs, which are listed here. So these are the sequence information, which are these motifs enriched in the specific gene of the promoter and may have some biological role. So next we screen all these motifs by electrophoretic mobility shift assay to see which one bind any of these, whether bind to a cyst protein. And we found that number 2 motif, which has is specifically bind to the cyst protein and may have some biological role. So we carried forward with this motif. So we named this as a insistence regulatory binding protein, insistence regulatory motif, and this motif specifically bind to cis nuclear extract. So what you are seeing here in the EMSA gel, the free radio level probe at the bottom, when it's bind to a protein, it gets shifted, which decreases with pole specific competitor 10 and 100x here, but not with non-specific competitor, which suggests that this binding is specific for particular motif. However, it doesn't bind to the trope nuclear extract, suggesting this protein may be absent in tropes, only expressing in the cyst. Next, we did some functional studies by using cis-specific promoter, which has either wild-type ERM motif or mutant ERM motif, and luciferase reporter gene and 3' regulatory region. So we make a construct and transfect into amoeba and make a stable transformant. And we did topogenoids, and in cyst, we did the luciferase assay. There is a significant reduction of luciferase reading in cyst, suggesting this mutation in ERM have impaired this function of promoter activity. And this ERM specific promoter activity is regulated through this motif. So the next question asks, we asked what protein binds to this motif. So we did a simple experiment by cutting this gel shifted band without the radioactivity and did the mass spec. And from the mass spec with biochemical validation, I'm not showing all the data here. So we find out this is a 29 kilodalton protein, which is annotated as hypothetical protein. And when we make a recombinant protein of this, it's able to bind to the ERM motif. And the RNA-seq data shows it's upregulated in cyst. If we look at the domain structure, it has a nicotinamide domain at the C terminus and a DNA binding domain at the N terminus. Overexpression of this protein with MIC tag so nuclear localization in trophozoites and also in coordinated cysts. Also, which support the notion of transcription factor, which are used to localize to the nucleus. Silencing of this protein significantly affect the insistence efficiency. So there is a significant reduction in cyst number when we silence this protein in amoeba. Not only the reduction of the cyst number, the cyst-like structure produced have irregular and defective cyst wall. So here, what you are seeing, chalcophore stain, which stain the chitin cyst wall, and the tophozoites don't take this stain. And here you can see in the control, it has a spherical and uniform staining, and most of the silence we do see irregular shape with a thin cyst wall, suggests that maybe some defect in cyst wall formation. With immunization with another cyst wall protein, that is Jacob, silencing of ermbp showed disintegrated localization of Jacob, which further suggests that indeed there is a difference or defect in a cyst wall formation. Next, we find another interesting metabolic factor that has role in encystation, that is NAD. NAD plays important role in many cellular processes like energy metabolism, lifespan, and DNA repair, and also transcription. So in many other reports, they have shown that the NAD level has a crucial role in this process. So we find that during the encystation, this NAD's level goes up. And addition of NAD into in vitro increase the DNA binding ability to the ERM. However, the NAD doesn't show any effect. Not only that, addition of extracellular NAD into the encystation media enhance the encystation efficiency significantly, which goes increase further when you overexpress this protein, suggesting, and however, it doesn't show any effect in silence ermbp, suggesting that NAD mediate increased encystation via ermbp proteins. So we further characterize different domain of this protein. So what you are seeing here, the alignment of protein sequence of ermbp in Entamoeba invading and Histolytica. There is a significant homology between these two pieces of this protein, and all the key residues which are involved either in catalysis, which are marked in blue, or catalytic and NAD binding marked as red. These are conserved. And here is the DNA binding motif. So we make recombinant protein, changing all these amino acids to alanine, and did bunch of biochemical experiment. So DNA binding was done by IMSA, as I mentioned earlier. NAD binding was done by protein thermal stability assay, and catalytic activity was done by enzymatic reaction, followed by HPLC and mass spec. So this is the summary of this our findings. So we find that ermbp wild type has all the functions like the DNA binding, NAD binding, and enzymatic activity. When we mutate this D12A and 150A, these lost the catalytic activity, and the DBM, the DNA binding mutant, shows lost the DNA binding ability. However, the C198A, that single mutant, shows have the greatest effect on ermbp function. It abolishes both NAD binding and also the nicotinamide turnover. So we express this mutant protein in amoebas to see the localization and their function. So here what you are seeing is a very busy slide, but I want to draw your attention into points. One is the ermbp wild type, its sole nuclear localization, as also seen in D12A and 150A. However, in these two mutants, DBM and C198A, it's failed to localize to the nucleus. And when you overexpress these two protein in Entamoeba, is showed phenotype like the silent ermbp. It's making some dominant negative effect, suggesting that overexpression of this mutant protein may be sequestering the factors which require for ermbp functions, and we are seeing some dominant negative effect. Another interesting feature we do see during the encystation is that when we put the trophozoates in encystation media, which is low glucose or hypoosmotic encystation media, it forms aggregates. The aggregate formation generally have been seen between 4 to 8 hours. And some of the cells get fused to make a giant cells. It's called multinuclear giant cell. And also you can see the division of nuclear division happens as we see during the encystation. So here, this is the bright field at cyto11, the pointed, the bigger, the giant cell is pointed in red. The cyto11 is to stain the DNA, calcaphore to stain the cyst. And you can see there are a lot of nucleus over 80 to 90, and it form a cluster. And we find that ermbp has a role in giant cell formation as well. So here you are seeing the control cells and the silencing or the overexpression of these two mutants significantly reduce the giant cell formation. However, there is no change in the aggregate formation, which suggests that ermbp function downstream of this cell aggregate formation, which is between 4 to 8 hours, and ermbp function downstream of this one. So another observation, we have seen that ermbp has conserved biological process in heat shock response in both Entamoeba, Histolytica, and invading. As I mentioned earlier, that Entamoeba, invading, and Histolytica has quite significant homology in this protein, and overexpression of this protein make parasite more resistant to the heat stress. So basically, this is the viability assay in heat stress and ermbp and control cells in heat stress. And also another phenomenon, we find that NAD level, NADH level ratio that goes up due to the heat stress. So there is a link between encystation and heat stress. And also heat stress induced the formation of quadrinucleated cyst, which can be seen overexpression of ermbp. Here you can see the MIG-tag, the quadrinucleated cyst in a field, which can be also seen in the wild type as well. But the percentage we have seen much more when you overexpress the ermbp. So with that, we came to a summary that there is a significant overlap between encystation and heat shock response. So in encystation, trophozoates, when we put into the encystation media, it forms aggregates, the NAD level goes up, ermbp function downstream of aggregate formation. We get three types of cell: mature cyst, multinucleated giant cells, and the trophozoates. In the heat stress response in Histolytica, we do kind of similar pattern. So it forms also aggregates, NAD level goes up, and ermbp function, and it gives quadrinucleated cyst-like structure and multinucleated giant cells. So with this, we came to this proposed model that ermbp directly bind to the NAD, which facilitates its bind to the transcription site and regulate the transcription of C-specific gene, which control the encystation. We also have find out that it has catalytic activity. It's convert nicotinamide to nicotinic acid, and this nicotinic acid may have some second messenger effect in the encystation. And also, we found that NAD level goes up during the encystation. So it's also possible that this nicotinic acid is used to make more NAD in through the sulfate pathway. So these are the caveats still need to be answered. So we don't know how this protein is regulating, whether it's binding to the RNA polymerase directly or some other factors is still to be explored. We don't know about the target genes, how what type of genes is regulated by ermbp, and also whether is the salvage pathway involved to make more NAD during encystation. So in conclusion, we identify a novel transcription factor, ermbp, which involved in amoebic encystation. NAD binding directly regulates ermbp function, and we identify significant overlap between encystation and heat stress. So future detection will be identification of target genes by defining binding sites of ermbp on genome-wide scale by ChIP-seq and RNA-seq. Identification of interactive partners of ERB also interesting by immunoprecipitation and mass spec. And this protein can be also used as drug target to prevent the transmission. So we talk about one lab at Stanford. So they have 700 compounds which barely reactive, very active. So these bearing variety of cysteine reactive electrophiles, meaning that they only interact with the cysteine and bind covalently. So we can screen that library in future. So with that, I want to thank my PhD supervisor, Professor Anurag Aloia at Bose Institute, where I start working with amoeba, and she's my biggest inspiration in amoeba biology and continue the research in amoeba field. And I also want to thank my postdoctoral supervisor, Professor Upinder Singh, and all the lab members like Gretchen, Danny, Hanwang for their contribution, Chris and Brett from Bojo Lab for the biochemical assay, Chris and Ryan from the mass spec facility for the mass spec, and for funding this work by NIH, and the following work will be with the Ramalingam Fellowship. And thank you all for your attention, and I'm happy to take any question. Thank you.
Thank you, Dr. Manna, for such timely and insightful talk. We really enjoyed it. May I request Shubho Motiva to take over and moderate the question answer session?
Sure. Good. Thank you.
Yep. So great talk, Dr. Manna. From the on behalf of the organizers, I'll quickly ask question if there is any question from the from the faculty members who are here, from the DLS faculty members who are here in the Google Meet session. First, I'll ask question from there, and then I'll take questions from the participants in the YouTube. So, is there any question from the faculty members here?
Yeah, I have a question.
Sure.
Hello. Am I audible, Shubhro?
Yeah. Shubhova, please go on.
Yeah, thank you, Dr. Manna. It was a very interesting talk. Coming back to the mutant study that you have done, you had said that there was a separation of function allele. So the two function, that the catalytic function and the DNA binding were separate.
Yes.
Then in one of the mutant, that is the C198 single amino acid change, resulted in complete abrogation of DNA binding as well as the catalysis. So I was wondering whether it distorted the structure, whether you looked at the structure of the protein, whether it was a general loss of structure due to the mutation, or was it really a kind of meaningful mutation?
It's a great question, and it's actually reported earlier, like in a JPC paper in 2008, I think. So it's mentioned that this C190 is involved in the D12, K150, and C190 make a pocket. So it has both the NAD binding and nicotinamide activity. So as you said, it's really good suggestion to look at the structure. So my previous lab, they're actually doing a cryo-EM for this protein to carry forward, to look at the structure and how it interacts. Yeah. So, but now we don't know that information. We don't have that.
Okay. Thank you.
Most welcome.
Okay. Any question? Next question, please.
So yes, I'm Chandra.
Chandrama. Yes.
Dipak, it's a
Yes.
great work. So I have one question that when you have done those study with the promoter binding,
Yes.
do you have have you done any kind of time point experiments? Like I want to ask that how long this particular protein is interacting with the promoter, like is binding with the promoter to regulate this entire conversion process.
Mhm. Yeah, we actually didn't do, mostly we did like 8 hours time point and 24 hours. These 8-hour time points, actually, we didn't see the expression of from the RNA-seq and also the quantitative RT-PCR. We didn't see the expression of this ermbp. It actually comes later time point after maybe 2, but we didn't actually dissect to 8 to 24 that time point. And we don't have exact answer like what particular time point it's expressed or how it's regulated. And another thing, we also find that it's also regulated with the function is dependent with also the availability of innate that also goes up during the institution process. We found that NAD level actually goes up little early, like at 8 hours, we see like almost twofold enrichment of the NAD level. But the ermbp actually come later. Yeah.
Okay. So and another thing, like I'm just curious that
Sure.
have you noticed any particular genes to be down regulated during
Yeah.
that 24 hours?
Yes, yes, yes. As a proof of concept, actually, as I said, like we found almost like 130 because we choose these specific genes, right? And we found that 130 genes, 31 genes has this CA that motif in the promoter. So as a proof of concept, we took like 3 or 4 genes, I believe, and when we overexpress ermbp protein, the genes which not regulated in TOPs, they are actually upregulated. And also when we silence this ermbp protein, the genes which upregulated during institution, those are not. So in that way, as a proof of concept, we have only very small data set, like only 4 proteins we tested. But we don't have the global idea or what are the genes in globally regulated by ERMP. We don't have that information yet.
Okay. Thank you.
Most welcome. Thank you.
Okay. Any more question from the audience in the Google Meet? Okay. If not, then I'll quickly take the questions from the participants who are in the YouTube. I mean, there are a lot of questions. I cannot take all of them for the sake of time, but I'll try to cover most of it. So Dipak, quick question from Omlan. The question is pretty interesting that because this is a human parasite, is if parasite's genome get updated when host genome is evolved. Do you have any comments on it?
So yeah, I mean that's the evolution. Like it's nothing is constant, so it's changing. So but it's now from the amoeba we previously annotated data, like which are like few years back, and now there are more information is coming, and we can see those changes if we compare, the previous data set, and everything is available in amoeba. If we look at the previous the genome or the genome sequencing and the recent data set from Neil Hall from UK, we can see that difference. Yeah, we can see some changes in the genome. Right? That's true.
Great. Okay, now pretty similar question from 2 participants. These are again very general question from Subhashish Indro and another from Spondon. That how Entamoeba species can cross the blood-brain barrier during infection, and how they invade or evade human immune response.
Yeah, so that's really great question. So one thing, so it has 2 questions: how it's entered to the bloodstream. So as I showed in that slide, like it has the the amoeba pore, those are cysteine protease, which actually virulence factor, which which release from the parasite and which make pore. Amoeba pore actually comes from the name. It has ability to make pore formation in the tissue. So it infects through the tissue and it can easily enter to the blood cell. And another characteristic for amoeba is if you get the amoeba from the patient sample from the colon, you will, if you stain, you can see a lot of red blood cells. So it actually engulfs the red blood cell. Red blood cell is comparatively smaller, and it's actually going through the phagocytosis process. You will do a lot of phagocytosis. So it can invade through the tissue, and it can mix to the bloodstream, and it can go to the brain. These are the quite rare to the brain amoebasis. The liver amoebasis is very common. We can think maybe it's close proximity from the intestine, large intestine. That's maybe one reason. Brain amoebasis is rare, but it happens. Yeah.
And so the other part is that how Entamoeba species can evade immune response.
Yeah, I actually missed that one. So amoeba has a very like smart technique. Like amoeba, amoeba recognize the galactose or galactosamine, N-acetyl galactosamine of the receptor protein to bind the host cells. Amoeba also has like glucose or galactose containing protein in the surface. So if you have suppose we have antibody which can human antibody which can recognize to the surface protein, amoeba has a technique to extrude extrude those antibody at the rear part. It's called and extrude into the media. So it has also been done in laboratory condition, like if we put the lectin antibody with the FIGC stain, and you can see after a certain time or if you can do the time lapse, you can see the amoeba extrude that antibody with a ligand and release it into the media. So that is one way to avoid the post-immune response. Yeah, that is one way, I think.
Okay. Now, one quick technical question from Onusha. That can this CRISPR-Cas9 technology be used in genetic manipulation in Entamoeba species? I think that you would love to see that, but I don't know the answer.
Yeah. I mean, it's a really relevant question, and everybody is using the Cas9 system. So this is the OP's lab actually did that experiment, like try to develop that CRISPR-Cas9 system for gene editing. And this paper is under review now in International Journal of Parasitology. So basically what we did here, we express, so the problem was to express the Cas9. So Cas9 is toxic most of the host cells or most of the cells. So we use the DD Cas9, like destructive box DD domain, which in that way we can express or stabilize that protein at particular stage we want. By adding TMP. We validate that one, and we have, from the preliminary data we have, the paper we submitted, we have preliminary data that it is doing the repair in the plasmid, but we didn't went further to see in the genome level yet. Because many other lab is also doing this CRISPR-Cas9, and we just try to publish this one first, so we submitted that one. So yeah.
Awesome. So a question from Abhishek Bardhan. He asks that any kind of change in subcellular localization of this transcription factor that you talked about was noticed during different stages, during the stage conversion, I guess, trophozoite to cyst, and do you see any change in subcellular localization? I think you have shown the data, but
yeah, I have shown the data. Mostly, I mean, we do see some stain in the when we stain in the trophozoite, mostly in the nucleus, and also certain in the cytosol, certain portion of the cytosol, and in sometime in the cyst, in the mature cyst, we exclusively see in the nucleus. So that is only the difference sometimes we notice, but there is not any other kind of localization pattern we have seen.
Okay. Diksha asks that if there is any relation between substrate availability and the ermbp expression.
Substrate availability and ermbp expression. It's a great question. We actually don't know, but the thing is, we know that ermbp is regulating the genes which are required for stage conversion and regulating the expression. But we actually don't know how ermbp is regulating. So in another project, so we are now truncating the promoter. Like we generally for the amoeba promoter analysis, we take 500 in nucleotide upstream. So we are actually truncating that 500 into different parts and analyzing this reporter construct to identify how this ermbp is itself regulating. So we don't know that one, but as for the substrate availability, substrate means we believe that it's bind to the promoter and recruit the RNA polymerase somehow, some way, and do the transcription. But rather than that, we don't have much information.
Okay, I think there is time for maybe one or two questions. I'll go quick. So this is the question from Shoma. The question is, we all want to know that if you are planning to develop any drug to block the ERM site, what will be your strategy, or maybe is this the strategy to block the ERM site for blocking the stage conversion?
Yeah. So that's the proposal. Actually, we actually proposed that one. So basically, so now we dissect some of these key residues which are important, and we have seen that this using recombinant proteins we can do this experiment. So the idea is we can do the protein thermal stability assay using a particular drug, or can screen the drug in 96 oil format. So protein thermal stability assay, I actually didn't get a chance to explain that one. So what it does, so if a protein bind to a ligand, it's stabilized. And if we denature that protein with the time course in a real time, so we can get a shift. So it stabilizes this protein, it's shifted little later compared to the wild type or without any ligand. So in that way, suppose we have 700 compounds we proposed. So we have those compounds, we can put those into 96L format and add the different drugs, and to see if any of this is binding to this wild type first, and then we can test with the C198 mutant. And in that way, we can shortlist it, the compounds, and which can be tested into the cells to see whether that has role to prevent the incitation in
Awesome. In the similar line of drug generation, there is one question that you have talked about the heat response, so heat shock response. So the question here from Shomo Kumar asking that if specific HSPs, what is the role of specific HSPs in heat shock response, and if they can be the target.
Yeah, so interesting thing is like, the incitation in laboratory condition we do with calorie restriction and or osmotic stress. So it is stress and also calorie restriction. So that's why you need to start certain genes and need to turn on some of the genes required for this particular stage. So that's why heat response, the HSP90 is actually reported in Entamoeba. The chaperone protein is negatively regulated the genes which need to be shut off during the incitation process. So the role for the ermbp, we found out that it correlates with the overexpression of ermbp, and it's the heat response, which is actually it's not the first time we are reporting. There are other labs like John Samuelson's lab, and also the Sudip Ghosh in 2008, they published a paper where they have also shown that there is a correlation between the heat stress and incitation are related. So they have shown that the proteins which are involved in cyst wall formation, like Jacob and chitinase and the topoisomerase 2, those genes which have role in incitation, they also are regulated due to heat stress. So as for the chaperone, so chaperone is actually negatively regulated, but there are quite supportive evidence that heat stress and the incitation is somehow related.
Fantastic. I think this will be my last question from the participants, that how altering gut microbiota provides protection against Entamoeba, species or proliferation.
Mm-hmm. So that's a really great question. So basically, you can consider like 500 million people are infected, are infected, but only 50, like 10% are symptomatic. So most of us like went through maybe some tasks in some of our lifespan. Sometimes we get this infection or dysentery. So most of the cases it's became asymptomatic, like 90%, as I said. So only 10% gets symptomatic. So most of so there is a correlation with the gut microbiota that actually protect the mucus layer, so amoeba don't get access to our yeah, uh, the mucus layer to invade or do the pathogenesis. But it's also true there are some the microorganisms or bacteria which actually enhance the infection, like P. pyrovim, that bacteria which enhance the infection of Entamoeba. So which is isolated from a cohort study from Bangladesh, like almost 100 patients from the children. They found the correlation of Entamoeba and the gut microbiome, the pyrovim, which actually facilitated the infection more. So yeah, I think there are some good microbiome biota and also some bad, which has both kind of roles.
Fantastic, Deepak. It was really nice to hear from you. I will have a quick question. Well, this is not really related to the talk, but it is like asking the question that why ancestration is so difficult to do in Entamoeba Histolytica. While we know, and you already said that you did everything in invading. So why it is so difficult? I mean, is there any known reason?
Yeah, so one thing is like from the previous studies, like if you isolate the Entamoeba from the patient and put into some in-situ media, you can get certain number of cysts, but that number is very low. But for prolonged study or prolonged, culture in the axenic culture in the laboratory condition that lost that kind of stage conversion abilities. So that is one reason. And recently, actually, it's not published yet. So people have make a model, like one lab at Wisconsin, USA. So they gavage the Entamoeba histolytica into a mice, and they have seen the cyst formation. So in laboratory condition, in vitro as such, we don't have any. Still, it's challenging. But if we have some model system or model animal, if that is useful to get cyst from histolytica, it will be very straightforward, and that will change paradigm of the research in stage conversion or stage developmental study in Histolytica.
Okay, fantastic. I really thank Dr. Manna for a fantastic talk. It's really entertaining to hear the stories of Entamoeba histolytica. And I thank on behalf of the organizer, I thank Dr. Manna. And also I thank all the participants for participating and raising fantastic questions. I'll just have a quick note for the participants who are in YouTube, that please don't forget to give feedback. The feedback link is there with the YouTube link. It will be activated only after 2 p.m. Tomorrow. So please either copy this link or come back to this link, YouTube link tomorrow and after, and please provide the feedback so that you can also get a e-certificate. So with that, I once again I thank Dr. Manna for his fantastic talk, and I guess I'll end this session, and I'll actually hand this to Sushmita. Sushmita.
Thank you, Dr. Manna, and thank you, Dr. Mujumdar. I am ending this scientific session now. So we will be here again at 1 p.m.
again.
Thank you.